Good morning, everyone, and thank you for joining us today. It is a pleasure to introduce our speaker, Dr. Jason Yeatman. Dr. Yeatman is an associate professor at Stanford University, where he leads the Brain Development and Education Lab. The goal of his research is to understand the mechanisms that underlie the process of learning to read, how they differ in children with dyslexia, and how to design effective literacy interventions across the wide spectrum of learning differences. Today, he will present his talk, From the Lab to the Classroom and Back, the virtuous cycle between education and neuroscience. In his talk, Dr. Yeatman will discuss how educational intervention offer a powerful tool to understand how a child environment shapes brain circuits for learning new cognitive functions, such as literacy. He will first present studies on the link between brain plasticity and learning to read, and then introduce the rapid online assessment of reading, a tool that enhances classroom reading assessments while providing new insights into the learning process. Please welcome Dr. Jason Yeatman.
Let me get closer to the microphone. I was just moving this. I don't know if anyone has to do that. It's like very hard to stay focused. Oh, there we go. That's better. Perfect. Okay. Well, I'm thrilled to be here today. I think we have an hour and 15 minutes. Feel free to raise your hand and ask questions as we go. I don't mind interruptions, but if it gets like many interruptions, then I might bring us back on topic. But yeah, feel free to ask questions along the way. I'll just get up and call on you. And I appreciate the conversation. So I'm going to start off giving a high level on my lab's approach. My lab does research at the intersection of education and neuroscience. And the overarching goal of our work is to inspire a virtuous cycle between these. I'm a firm believer in interdisciplinary research, and that by spanning multiple disciplines, we're poised to make discoveries that aren't possible within the typical confines of, for example, education or neuroscience. So let me give an example. So now, as of 6 years ago, I moved from University of Washington to Stanford. My primary appointment's in the Graduate School of Education. And many of the studies in my lab start off looking like a typical study you'd see in a school of education. So small group reading instruction or reading intervention programs, often a teacher working with a small group or even one-on-one with young kids. And as a neuroscientist, I see this educational approach as a powerful tool to ask basic scientific questions about brain plasticity. So through educational interventions, we're able to run randomized controlled trials, have tight control over different aspects of a child's environment, then make measurements of how those changes to the environment induce plasticity in brain circuits. So throughout this talk, well, by the end of the first part of the talk, this figure, which is not just a picture, it's actually data from this study. I'm going to unpack what we're actually doing. Now, the long-term goal is to think about closing this cycle, thinking about neuroscience as a tool to predict learning and personalized education. Sitting in a school of education now, it's often thought that we can look under the hood and know exactly what's going on for each kid. All the people that study brains here will know that that's not actually the reality of MRI-based research now. But I think for many of us, it is a long-term vision that through measuring individuals' unique developmental trajectories, that we're going to be able to glean more information about how that individual's brain's developing and think about a personalized approach to intervention or education that is tailored to their unique pattern of brain maturation. Now, that's an aspiration, not a current reality. But one thing that came out of this research, and this is where I'm going to end my talk, is on the rapid online assessment of reading. This is a assessment platform that sprung from this research when we were all secluded at home during the pandemic, and now it's grown to be the foundation of most of the work we're doing. Actually brings a lot of the findings from our research into schools, and then through collaboration with schools, inspires the next generation of research questions. Okay, so for those of you that don't think about reading all day, I want to start off by making the example of how unique literacy is in the context of human development. So when I look at this page and this now outdated photo where I had a little more hair and my daughter was completely illiterate, I can't help but hear the sounds of language that are written down on it. As I look at the page, through my eyes, I'm seeing, my brain is encoding the symbols written down by the author, and I'm experiencing it as language. Whereas my daughter is looking at the pages, seeing pretty pictures and squiggles on the page, right? The letters that are written down at this point, I think she was almost a year old, mean nothing to her. And unlike other aspects of human development, the two people in this photo see fundamentally different things for many years to come. So unlike other aspects of language, for example, so here's her months later, where she is suddenly able to communicate and demand that I read her more books or do many other things, but still, so at this point, we haven't taught her anything per se. She is learning how to produce language and understand language just from being in an environment where her brain is made. No one has to teach her the series of intricate. Okay, so even as years come, so this is her then with her younger daughter, or sorry, my younger daughter, her younger sister. At this point, she was about 4.5, still completely illiterate, but an incredible communicator in spoken English. She can pretend to read books to her younger sister. She can talk. She can communicate. Many aspects of child development progress in a continuous manner, right? There's not there as a parent, don't observe major discontinuities in her learning to walk and talk, speak, communicate. Yet at this point, 4.5 years old, she's a proficient communicator. Yet the squiggles on the page still mean nothing to her young brain. Now, this was taken at the end of kindergarten, so about a year after this photo, and now she has begun to unlock written language. She actually now is starting to teach her younger sister, taking some burden off of us and teaching our younger sister how to actually produce written language. So the point I want you all to have in mind is that literacy is an invention of modern societies. The human brain is not pre-wired to learn to read. Unlike spoken language, no amount of books piled in front of a young child is going to teach them how to unlock communication through written language. It takes years of instruction and practice, particularly for a complex written language like English, in how to map symbols to sound. And so the neural circuitry of literacy is constructed in response to education. And thinking about that first slide, what I often argue is that literacy is a model system for studying brain plasticity. That we can run ethical RCTs, randomized controlled trials, where we manipulate aspects of literacy instruction and make measurements about how that sculpts the young developing brain. So let me give an example of it. Thinking about what's going on in my brain versus my young daughter's brain as we're looking at this book here. So of course we're first opening the book, looking at the first picture, in many ways, the first set of computations our brains are performing are quite similar, even at this young age for her. So of course, information's encoded by the retina, sent back to the very back of the brain, primary visual cortex, that's starting to encode the simplest features of the visual image. I could give another talk going through this part in detail, but we think of like a series of operations the brain is performing. Primarily is observed by circuits along the ventral surface of the brain that perform a series of computations that transform these visual features into the recognition that we're both looking at a picture of a bunny rabbit. Now, in my brain, as a 30-something-year-old there, and my daughter's almost 1-year-old brain, many of these computations are quite similar. Maybe a couple hundred or 100 milliseconds slower in her brain, but for all intents and purposes, the brain is computing much of the same information. Now, of course, things are fundamentally different in my brain as soon as my eyes saccade to the text on the page. So within this broad circuit along the ventral surface of the brain that's involved in visual recognition lies in my literate adult brain a specialized region for visual word recognition, a region that is the interface between our visual system and our language system. You can think about this as a region that develops as a function of learning to read and provides this interface between circuits that evolved for visual recognition, circuits that evolved for spoken language, and through the process of learning, we craft this new region that spans between these to evolutionarily older circuits, allowing this new cultural invention of literacy to be unlocked in my brain. Now, of course, this region, coined by scientists as the visual word form area, is of course not specialized for reading in my daughter's brain, as at this point she has yet to master literacy. But of course, I'm not just leaping through the pages, I'm reading out loud to her. And in her brain, as I'm reading out loud, the spoken language circuits are becoming active, performing many of the same similar operations as my spoken language circuitry, but without this go-between between visual and language. So a way I often think about literacy is that it's plugging the visual system into the language system in a new way, that it's allowing information to flow rapidly between circuits that evolve for different purposes and connecting them in a way that allows for a new cognitive function, literacy. So when we think about it this way, of course, the white matter pathways that underlie this circuit are critical. So if we dissolve away the cortex, look at the connections beneath the cortex, information needs to travel rapidly between visual and language circuits. I'm going to be highlighting two key pathways at the talk. The names aren't important though for those that aren't familiar with the anatomy, but the arcuate fasciculus shown here in blue, the key connection of the brain's language circuitry. The inferior longitudinal fasciculus is a major output of visual cortex, allowing information flow from early visual regions, primarily up the ventral stream. So 4 points to keep in mind throughout the talk. First, that learning to read drives the development of specialized brain regions for rapid word recognition. The analogy I gave is that what literacy means is that written words are plugged into the systems that evolved for spoken. Connections between visual and language circuits are essential to allow for rapid information transfer between these regions. And finally, in the literate brain, written words are processed like spoken words. So once you progress outside of visual cortex, in all of your literate brains, the information that is being processed by your language circuits as you're reading this is quite similar to the information that's being processed were I to read the text out loud. Then, of course, brains are different. Learning to read is easy for some and difficult for others. This is what we refer to as developmental dyslexia. So when I made this point that in the literate brain, written words are processed like spoken words, another way to think about this is that once you become an expert with literacy, it's automatic and unconscious. You don't have to think about decoding the text on the slide. It's an operation that all of your brains are performing, whether you want your brain to or not. So let me give you a quick sense of how we can run experiments to look at that. So we recently ran the one and only MEG study that I'll probably ever run. Do you guys have MEG here? Okay. I have a contentious relationship. I'm very excited about this study. So we wanted to get at measuring the automatic stimulus-driven neural response to text. So what do I mean this? We're showing a stream of words and pseudo words and then matched visual patterns that have no text. Okay, so the stimuli, you see a word or a pseudo word. Sometimes you see a patch of noise that's matched in terms of the spectral frequency of the visual image. And on this same stream, you switch between two tasks. One task, you're doing challenging psychophysical judgments on the fixation dot. People always say that judging color of a fixation dot is easy, but you can actually set it up with a staircase to make it quite challenging so that all your attention is devoted to making judgments of the fixation dot and ignoring the text you're seeing. And then other points in the task, or the stimuli remains the same, but we now ask you to read the text and make a lexical judgment. So what we did in the study is we used the lexical decision task to first define brain regions that are sensitive to text. So basically that respond when you're actively reading the words. So this is showing the contrast between words versus noise patches while participants are performing the lexical decision task. For those here that study language, you'll see the kind of canonical language circuitry becoming active as it's a language task. And what we could do is we can define a particular region. So in this case, the superior temporal gyrus, this portion of the superior temporal gyrus that's becoming activated early on as participants are forming this lexical judgment. This is a region that's canonically involved in processing the spoken sounds, like the sound structure of spoken language. And we can now ask what this region is doing when you're still seeing the text, but you're performing an irrelevant task where you're focusing on the fixation. So this is showing that analysis of the fixation task in children with typical reading skills, so those that are effectively mastering literacy, versus those that are struggling to master literacy. What I want you to appreciate is that in typical readers, and you would see the same thing in adults, that as soon as a word is presented on the screen, irrespective of whether you're trying to read it or trying to ignore it or trying extremely hard to ignore it by doing a separate psychophysical task, you still see a larger response in this language region to text compared to the visual control condition. But you don't see this in the struggling readers. Look at this in a different way and look at the selectivity, so basically how much this region responds to text compared to noise patterns. And so that's shown along this axis versus participants' reading skills on the y-axis. You can see a nice correlation between the two, showing that as you master reading skills, this transformation from visual information into your language system is performed even against your own will, whereas this automaticity is not present in children who are still struggling to learn. Yeah. Yeah, good question. So, so this study, one of my many gripes with MEG. So this study was really designed actually to look at specific computations of the visual word form area, and this was a side result that that we thought was nice from here. A lot of the activation along the ventral surface, you have a lot of variable sensitivity with MEG, and so what we can't figure out in all the studies, of which there's a number that we've run looking at MEG responses to text, there's a lot of variability and localization along the ventral surface due to distance from the MEG sensors and various things. And it never get, and this is like different than what other labs see. I don't know, there's probably MEG experts here who will tell me that if I had used a different source localization algorithm, everything would have looked better. But that was one of the reasons I moved into the MEG world and then back out, is that changes to the source localization algorithm would have dramatic effects on where you do and don't see activation. So we stuck with the simplest approach that we felt confident in. Like the boring answer to the good question. Okay, so that's what I mean by literate words or process like spoken words, that text is transformed into the language system, even when you're trying actively to know it. I think this is a hallmark of. Okay, so now I'm going to get transition to thinking about plasticity in children. And I want to start off with the definition of dyslexia that came from Europe. I'm not going to make bad jokes about what's currently going on in the National Institute of Health in America, but we used to run panels that would come to consensuses about definitions. That's apparently going out of the wayside now, but the dyslexia is a specific learning disability. This was a key part of the definition that was added in the early 2000s that identified learning disabilities as being neurobiological in origin. This was a large push from parent advocacy groups and also evolving science that was showing the differences in the brain that were associated with learning disabilities like dyslexia. And tracking this over the next couple decades, it's had this change to the definition has had some benefits, people appreciating that learning disabilities are a real thing, that some students struggle to learn due to differences in their brain. But the definition has often been interpreted to imply that children with dyslexia have a neural defect that causes persistent difficulties with reading, irrespective of intervention. And when we look at the research base for this, we can note that there's a wealth of research on differences in the dyslexic brain, but far less on the relative stability or plasticity. And I'm guessing there maybe are a couple people in the room that are interested in reading and dyslexia specifically, probably a broader room full of people that are interested in other aspects of cognition. I'd like to make the point that I think this is true about cognitive development in general. Whatever function you are personally interested in, whether it's executive functions or working memory or differences in emotional regulation, we have hundreds or thousands of studies that do group comparisons to look at differences among groups, or correlational studies that look at the correlation between some aspect of the brain, some aspect of behavior. Maybe 1% of studies are longitudinal, tracking these differences over time, and maybe like 0.1% actually form interventions to ask how stable is this. So this has been a primary goal of our research over the last decade, is examining to what extent the phenomenon that we talk about when we characterize the dyslexic brain are a true intrinsic trait of these individuals' brains, or to what extent they reflect differences that evolved over time due to differences. So what do I mean by this? Let's think about one of the most well-reported differences in the dyslexic brain. So I made this point early on, pointing to white matter connections. So these bundles of axons that connect together the brain's reading circuitry. It's clear, I don't know, I would say for decades, but actually for centuries since Wernicke's original work in the 1800s, that the white matter is critical for many high-level cognitive functions, particularly for reading, where information must be rapidly transmitted between visual, language, auditory processes. One of the most reproduced differences when people look at children with dyslexia, or look at the relationship between differences in brain anatomy and reading skill, is that the structure of white matter connections is correlated with reading skill. So actually the first data my lab collected when I first started my first faculty position in Washington. So I'll unpack the measures more later, but this is showing a correlation between a measure of water diffusion based on diffusion MRI and children's reading skills within the arcuate fasciculus. And this is was a replication of like a dozen or so other studies. So you look at it as a correlation or a group comparison. This particular pathway, the arcuate fasciculus, has a long history of being associated with differences in reading. So we might ask, and I think many people look at this, I'll note, across the many brain behavior correlational studies, we're all good scientists. We know correlation isn't causation, but most of us put our brain measure on the x-axis and our behavioral measure on the y-axis with some kind of loose assertion of causality. We tend to think about brains as being more fixed and behavior as being more malleable. So many people look at a plot like this and make the assumption that these white matter properties, the properties of the brain that we're measuring, are a static trait that is causing differences in learning over time. So if we think about this plot, I think this is the model many people have in their mind, is that you have participants that are up here in the plot, you have participants that are down here, and of course the brain is developing, but likely over development, these groups of participants probably remain different, right? That would be the idea of that there is a static trait associated with dyslexia. Alternatively, we can ask if instead these differences in brain properties really reflect the effect of differences in experience. If it's differences in environmental factors that are driving participants to be at different points on this axis of brain properties. So what might this look like? We can imagine there's some kind of normative developmental curve for the white matter. We imagine an individual progressing on this curve. Maybe they're born and they start along the typical trajectory, and they're in a fancy place like Palo Alto, and they enroll in a really good preschool, and maybe that gives a little bump to development. But then maybe they enter kindergarten, things are going well, but over the course of the year, they find themselves struggling, unable to connect with their teacher and peers, and maybe this experience in the classroom is actually a cause in creating their a abnormal developmental trajectory. Then in the United States, in wealthy, high-resource places like Palo Alto, in this scenario, parents often pull their kids from a public school, enroll them in a fancy private school, and maybe this is a factor that fundamentally changes this individual's developmental. Of course, so under this model, what the implication here is that what we're measuring at a given time point is not reflecting an intrinsic property of this individual's brain, but it's instead reflecting properties of the state that that brain is developing. So properties of that child's immediate environment as opposed to something that is intrinsic to their brain over the course of development. Now, of course, this is not an either or question. We're not asking, is the brain plastic or not, or is it nature or nurture? It's really a question of effect sizes and time scale, right? We know that experience influences development. We also know that genetics lay out a lot of the scaffolding of brain development. And here what we're asking about is the time scale and effect size for how much changes in the educational environment are going to resculpt white matter development. So here's the present study. We're going to look at this moment. We're going to impose this change on kids, and we're going to measure how that's affecting brain development. So the study designed to ask this question about relative stability or plasticity of the brain's white matter. We recruited 34 children with dyslexia for an intensive summer reading intervention program. I'm not going to go through the details of the intervention. Happy to talk about interventions or offline. But I bolded the most important part. So it's a very intensive program. This is kids coming in for their summer. They're spending 4 hours a day, 5 days a week for 8 weeks doing one-on-one training in the skill that is the most challenging to them, reading. And it's not just practicing reading, it's going through a sequence of a curriculum that focuses on like kind of rebuilding the foundations. Then we also had a control group that was going through business as usual, so just being scanned longitudinally over the course of their typical school year. And kids came into the lab along with the intervention program at 2-week intervals where we collect behavioral measures to look at improvements in academic skills, then also collecting brain imaging measures with diffusion MRI. Don't worry, I'll unpack the methods more when we get to the relevant. Yeah. During the school year. Yeah, and so that an important point is this was not a randomized controlled trial. So I'm going to be using causal language throughout. I have a number of reasons to believe that the effects I'm going to show are causally driven by the intervention, but there are other interpretations, right? And I'm totally open to other interpretations too. One of the reasons for this is like when you're doing an intervention that's this intensive, it wouldn't be ethical to run it as an RCT. I couldn't ethically ask a family to give up their whole summer vacation to do something that I wouldn't believe is effective for their kid. And in terms of like a placebo or other things you might do, like there's like you could look at different types of intervention programs. That wasn't the question here. The question here is like about this change in the educational environment. So I want you all to think for a moment what day-to-day school is like for children with dyslexia. When you have kids that have been struggling for years, often every day of school is like, for many of them, can be a traumatic experience. You're being asked and put on the spot publicly to do something that every day in school that you just can't do, right? So in the US, at least, like the rate of antidepressant and anti-anxiety medication use amongst young kids is quite high, like over 50% for the population. Yeah, I saw a lot of shock cases. Right? These are like 8-year-old kids who are already being medicated for anxiety disorders, for major depressive disorders. And a lot of that's driven by the fact that school is traumatically difficult. Right? So for these kids, you end your school year, and then now for many of them, for the first time, they're working one-on-one with someone who is meeting them where they are and proving to them that they can learn this skill. Yeah, there's a question there. Yes, yeah, I'll ramble about that for 1 second. Yes, the goal here is not to say which intervention program is more effective or which component of the intervention is more effective. We were really trying to get at this question, like going back to the slide, like thinking about like what should our model be about how a change in your environment can impose a change in your brain organization. So for going for that question, what we're looking for is the biggest effect size that we can have, right? And so in this case, we chose an intervention program that we already know is quite effective and also has the benefit of being an incredibly intensive approach. I'd say I think this is also one of the benefits for those that are studying brain plasticity about thinking about educational intervention, is that it allows for large effect size studies. There are, you know, my background is in vision research, and I could not get a kid to come in and do psychophysics in a dark room in the lab for 4 hours a day for their whole summer, right? Like no parent's going to volunteer their kid to do that. But I most certainly can get parents to volunteer their kids to enroll in a highly effective dyslexia intervention. Then the other benefit is we get to offer them highly effective intervention for free. Yeah, any more questions while I'm, yeah, up front. Yeah, great question. 7 to 13, large age range. Okay, so here's looking at behavioral data. So these are 3 measures: reading, reading accuracy in orange, reading fluency in blue, reading speed in green. This is during a baseline period during their typical school year. These are all age-adjusted standard scores. So the population average would be 100. Standard deviation is 15. So these are all kids that are well below average. And standard scores are stable or declining during this baseline period, meaning that they're not improving over the course of their normal school year. School year ends, they enter the intervention program, and we see significant, roughly linear growth. So by linear, it means that for each hour of intervention, you're getting an incremental improvement across reading scores. And we see different effect sizes, but large effects actually ranging from cones D of about 0.5 to 1.2, depending on which reading measure you use. But the point being that you can see large effects. This is about a year and a half of learning over a relatively brief period. So we have large behavioral effects to work with when we think about now relating these changes in brain. So now we're going to get to our primary question of whether white matter properties dynamically change in response to intervention. Normally I'd spend a lot of time talking about software and methods. I'm not going to today, but I don't know. I have meetings with a bunch of people this afternoon. Happy to talk more. Open source software has been a major part of our research over the years. Now I'm going to really just highlight work by my friends and colleagues, Ariel Rokem and John Kruper, who are now carrying the torch and building all this software. To give everyone an intuition about what we're measuring, right? So I showed this rendering of the arcuate fasciculus. This is, a rendering based on computer software. This is what the pathway might look like if, under an electron microscope. So this is showing a reconstruction of nerve fibers based on electron microscopy. You can see that in many parts of the white matter, you have axons aligned and bundled together. And what we're measuring is the diffusion of water around these axons. So now we're looking at a simulation where we think about axons as tubes, water molecules as molecules that are moving due to random motion. And what diffusion MRI gives us is the rate of motion, rate of diffusion across different dimensions. Why is this interesting? Well, if you're a physicist, Brownian motion is just interesting. But if you're a neuroscientist, water diffusion is a way to probe the cellular structure of tissue. So in this case, where we think about a child's brain developing into an adult brain, so what's changing the white matter, axons are growing larger in caliber, so going from smaller axons to larger axons. They're growing more insulated, so additional wraps of myelin around the axons. In terms of water diffusion, these are creating more barriers to the diffusion process. So when we make a measurement of the white matter with diffusion MRI, we're going to see a decrease in water diffusion as tissue gets more complex. Why does this matter for computations? That as axons grow larger in caliber and more myelinated, we're able to send more information quicker. You can think about this change over development as being an increase in the bandwidth of the connection. Basically, more information to be sent per unit of time, the timing precision faster, and so on. So this is like the fundamental measurement we're making. We're going to be looking at changes in the rate of diffusion as a function of learning. How we make these measurements is through a software package called AFQ, or Automated Fiber Quantification. Takes raw diffusion MRI data off the scanner. You can fit a number of different models of the diffusion signal that characterize the distribution of fiber orientation in each voxel. Can then use tractography to build a connectome, basically of all the white matter connections that can be traced within this individual's brain under this model. We then apply anatomical knowledge to identify specific brain pathways in each individual. So here showing the inferior frontal occipital fasciculus segmented in this individual's brain. We then map different properties of the tissue. So for example, mean diffusivity or the rate of water diffusion, which was shown in the last slide, but there's many different properties that you can measure based on diffusion. So this is what the pathway might look like when you map on the values. And then we represent this as a vector of measurements going basically along the length of the pathway. You get the profile of diffusion properties along the length of each pathway in each participant. And this has grown into a ecosystem of tools that all operate on raw data coming off the scanner. Now like an online tool for visualizing and communicating data. For those that are interested or want to contribute to ongoing software projects, just want to highlight the work of all my collaborators on this. Number of kind of upcoming projects connecting fusion data with new machine learning methods and new visualization methods. So now let's look at how these pathways are changing over the course of learning. So we're going to look at the arcuate fasciculus. Here's diffusion during the first session of the intervention group. They come in 2 weeks after 2 weeks of learning, and we already see a highly significant decrease in diffusion. And the decrease persists or maybe it grows a little more over time, but we see the largest change between the first and second session, basically like right when they start the intervention program and are in this dramatically new environment. This is the control group. So we see a highly significant decrease in diffusion in the intervention group, no change in the control group. For those that aren't used to thinking about diffusion, I kind of looked at this, the cellular properties of what diffusion is tapping into. If you prefer an analogy to plumbing, you can think about less water diffusion as indicating better insulation. As we have like less diffusion, it's because there's more insulation around the axons. Or if you prefer an internet analogy to a plumbing analogy, this might indicate a higher bandwidth connection, though there are many other cellular interpretations of like what's actually changing. Okay, so excited to say this was recently replicated too. So the inference we're drawing, and I think this is a pretty solid inference based on the data, is that actually going into this new experience is over a very rapid timescale, driving the growth of a measurable growth of new tissue within the pathway. You can then ask, are these mechanisms of plasticity localized to, for example, just the arcuate fasciculus, or do we see them more broadly distributed? Well, we're going to look at these two pathways now. We're going to look at data for one individual. So remember, we have 4 measurement sessions. We've normalized the data to put measures from these two pathways on the same axis. And we're looking at the growth trajectory in terms of mean diffusivity for the arcuate and the inferior longitudinal fasciculus. What you can see for this individual is this rapid decrease in diffusion. This individual actually looks a lot like the group average, kind of levels out over time. Basically, you see the same trajectory of change across these two pathways that are separated by many centimeters. And look across individuals, and what you can see is that growth trajectories for these two very different pathways are tightly coupled. Fit a growth rate and look at the relationship between the growth rate of the arcuate versus the ILF, and they're highly correlated, meaning that we're seeing significant changes in both pathways, and those changes are occurring almost in perfect unison for these two different white matter pathways. And then expand out and look at a number of different pathways. So getting to the first figure from the talk, this is showing a variety of pathways in the brain, heat map based on the p-values from a model, a statistical model, looking at change in the intervention group versus the control group. What you can see is that plasticity is widespread, so not just localized to a single isolated brain region. These two bars were the ones that I showed you before. So arcuate and ILF, these are like the pathways that we had hypotheses about. They're showing highly correlated change, and what we see is actually correlated change within a network of pathways. Actually, there's a couple different networks that are changing over their own timescales, and happy to chat about that more later. We also now look at the link between plasticity and learning. And we can do the same thing of normalize the reading scores based on z-scoring each individual reading scores. We can now look at each individual's trajectory of change in terms of diffusion data in the white matter versus their growth in reading scores. The same first individual that shows a rapid decrease in diffusion also shows a rapid increase in reading scores that flatten out over time. You can see similar trajectories across individuals. We can now look at the relationship between session-to-session changes in diffusion MRI versus session-to-session changes in reading skill. What we can see is that the time course of white matter plasticity is tracking learning in individuals. Of course, there's also a lot of unexplained variance too. So statistically speaking, these are occurring in unison, but there are also improvements in reading scores that aren't predicted by improvements or changes in the white matter. Okay, so summary of results so far. So the interventions can lead to large-scale changes in brain structure that occur over a rapid time scale and in synchrony with growth in reading skills. I put the asterisk here because it's important to remember, like, we're working with a quite intensive intervention program. I talked a bit at the beginning about like the profound change that entering an intervention like this is for a child's day-to-day life. So the assertion is not that like every single thing you learn in school, like your brain is going to totally reorganize itself. But what we do see is that over this time scale, and this is a time scale of weeks, that white matter constitutes a highly plastic system that's changing relative to changes in the environment. So to give you a sense of effect sizes, when we look at effect sizes in terms of like how much change individuals show in their white matter, we see like Cohen's D of 0.5 to 1, so quite large effect size in terms of what's the capacity for change within an individual's brain. And when we compare that to doing a literature review and saying like, well, what's the average effect size you see when you compare dyslexic brains to typical brains, or choose your other cognitive difference. Effect sizes are usually about 0.2. So the takeaway point here is that we can see more change in an individual over a pretty short time scale than we would expect in an average group comparison. So I want to leave everyone thinking about that point. I mean, we can, there's many questions about what happens over longer time scales, but whenever you look at a group comparison saying this part of the brain differs in this condition, it's really important to think about how stable or malleable this difference might be. In this case, we're seeing that the structure of the circuit is much more malleable than we might have originally expected. By the way, what time should I go to? 15 minutes. Okay, cool. I want to close the loop, and should I take questions now, or should I finish up? Okay, all right, I'm going to take 15 more minutes, then I'll leave plenty of time. So I want to get to the applied part too. So this has been ongoing research over the last decade, and from this research we've invented the rapid online assessment of reading. Switch to a different slide deck quickly. So I'm gonna just take 10 minutes on this last part to talk a little bit about ROAR. And our high-level goal here is building bridges between the lab community and the classroom, trying to create a pipeline that allows for deeper connections between research and practice, really thinking about this as a two-way street, not just the classic tech transfer where universities invent something and then like bring it to society, but instead also bringing educators into the loop and giving schools the opportunity also to influence the future directions of our research. So as I showed in the last slide, and historically what my lab has done is study the interplay between brain development, plasticity, and learning. This involves the development of intervention programs, a lot of engineering work around brain imaging measurements and software, and of course, the development of assessments, usually using like fancy computer hardware and expensive eye trackers and a variety of things that work very well in the lab, but would not work in the world. If anyone remembers March of 2020, we were all sent home and sitting at home and trying to figure out how to do work over Zoom. And the first challenge that we sought to solve is that since we can no longer sit in a room one-on-one with a kid and administer standardized reading assessments, wanted to see if we could develop gamified web applications that get the same measures, not that anyone could take, from the comfort of their home at this point, and that could replace the time and resource intensive standardized. What came from this is the rapid online assessment of reading, or particular first assessment, which was a single word reading measure, basically trying to replace the measures that we would typically use for measuring reading skills, for example, in that brain imaging study I just. Where this has grown to now, so beyond the fact that I'll tell the quick story of how we were able to optimize this measure to actually be more reliable than the old standards it was validated against. The outcome now is that we're able to do research at a scale that I never really thought of before that. We're able to reach populations all around the United States, actually around the world, and also have much more inclusive research. So have research that doesn't just reflect the children of professors who happen to live around a university, but actually study reading development across the diversity of experiences around the United States and around the world to some extent. So the direction we're taking this is the idea of deep phenotyping, of trying to understand the collection of factors that drive an individual's struggles, as opposed to just studying like what is the one major challenge that you can detect in like the small n studies that have historically. Our method for doing this is through a research practice partnership model. What this means is that we're building measures in the lab, but we're working closely with schools and clinics all around the United States that are using, as we validate the measures, are using them standard practice, and also providing insights that are influencing the future directions of research. All the papers from this, we're trying to also bring the movement of scientific transparency to education. Education, at least in the United States, is like the opposite of transparent. Most products, it's like quite hard to find what is the scientific backing. So every paper we publish from this, obviously, is published in open access journals. Find them all online. ROAR is now expanded, so we're now in, this is our new map, I just put this in this morning. So we're in 29 states in the United States, in 5 countries. We've been approved as an official dyslexia screener, being used universally across the state, in 2 states in the US now. And so we're working on expanding to basically serving hopefully 1 million children around the United States, at least, through this research practice partnership model, which allows us to ask questions about longitudinal development and large representative samples that span an incredible diversity of learning. Okay, so what does the task look like? So we began by testing the hypothesis about a efficient and scalable way to measure single word reading. Reading's typically measured by having kids read out loud in a one-on-one setting, and having a clinician or educator like score their responses, for pronunciation and reading speed. And we had the hypothesis that a simple lexical decision task, text is flashed on the screen, your task is to decide whether it's a real word or not, could measure the same skill. Everyone's gonna presume, there's many people that probably use lexical decision tasks and are gonna say, wait, that's quite different than reading out loud. And yes, it is. But the hypothesis is that the underlying mechanisms of what the mind is doing are the same, and the constraints on reading development are gonna be the same across these 2 very different types of tasks. So here is our first proof of concept study. It's actually done right at the beginning of the pandemic. We had kids that came in for the next version of the intervention study. We had already assessed their reading skills, then the world shut down. So we had the reading skills measured one-on-one in person, created this first version of ROAR, which was long and simple. And we found across my career, like the highest correlation between 2 measures. So it's a good proof of concept that the approach might be valid. We then went on to do a series of studies where we then built like a faster, more efficient, better gamified version that was like clearer for younger subjects. We ran a new study now with this more efficient version of ROAR in 6 and 7-year-old students, individually administered the Woodcock-Johnson, once again found an exceptionally high correlation between the 2, and then moved on to start doing validation studies in hundreds of students around the United States. Found that these effects held up across different demographic groups, different locations in the US. So one of the benefits now of moving to online research is that we can now efficiently run studies at a much larger scale. So I have a student that's been building new computer adaptive testing algorithms, and they're all open source. For those that are doing online research, you can check out JSCat online here. So Anya ran an experiment with a series of collaborating schools where she deployed the standard version of ROAR versus one that used a new computer adaptive testing approach to basically like tailor the properties of the items to an individual's ability, and found that we were able to achieve above 0.9 test-retest reliability and about 40% fewer items. So by through this approach now, we can keep honing the assessment, making it more efficient, quicker, and more precise. And then the other part is, I know there's a lot of really cool research here along these lines, but she's studying the gamification and how creating engaging characters and like different aspects of gameplay can improve the precision of the measure. So that is a description of our first single word recognition measure. We now have built this out into a platform with a suite of measures that target different aspects of reading development. I'm just going to skip to finishing up here. So we have a suite of measures that kind of target the foundation of reading skills. Each has been validated through studies that start off in the lab and then expand to working collaboratively with schools around the United States, eventually abroad more, but capacity limits. So why do schools engage with us in this research? So we have engineered this into a platform that interfaces with all the student identification systems used around the US. So for a school to use ROAR, just requires a data usage agreement with the school, and the assessment now shows up on every laptop across the school. Kids can log in just by holding a QR code up to their webcam. They'll see the dashboard of games they've been assigned. And in real time, as students are taking the assessment, data is being written to a real-time database, and the teacher in the classroom is seeing the assessment data in real time, so able to see which students are flagged for having indications of dyslexia, and they get interactive score reports that are rendering basically as each student finishes the assessment. So a superintendent can look across the whole school district, or a teacher can look at their classroom, parents can look at their individual students. So through this, we now have the pipeline to bring new measures developed in the lab to any school around the United States, and also take feedback of teachers and school administrators about what directions they would like to see the research going. So one of the current directions is thinking about reading development in multilingual learners. Like a trivially obvious question anywhere in the world except the United States. I'm guessing every person in this room here is multilingual, probably. But one of the big questions is thinking about how bilingualism or being taught to read in the language that's not your native language interfaces with biological risk for dyslexia, and how we can actually develop new teaching methods that are more equitable for students that are entering the US school system speaking a language other than English, having to both learn English and learn to read it. The other goal, as I mentioned earlier, and very much interested in collaboration on this, if people here have particular hypotheses that we might be able to test. So when we think about students with reading disabilities, phonological awareness, or your understanding of the sound structure of spoken language, has been established since the 80s as kind of one of these fundamental mechanisms that influences reading development. Starting in the 90s, another dimension of reading difficulties was discovered, and kind of these two key factors, phonological awareness and rapid automatized naming, which is how quickly you can transform visual information into verbal information. Now kind of the foundation of most dyslexia research. It's written into most legislation. It's been studied across cultures, across every country. But there's many other aspects of cognitive development that there are theories that relate to reading development. So executive functions, for example, or even thinking about specific aspects of executive function. Actually, some great research from this university looking at that. My own lab has studied the relationship between aspects of visual processing and dyslexia. And what we're moving towards is trying to think about reading difficulties within this high dimensional space, rather than thinking about, what is the single cause of a child's struggles. Instead, thinking about, for example, the students who have exceptional phonological awareness and rapid onomatized naming, but still end up with a dyslexia diagnosis. Understanding what are the contributing factors that together interact with the learning environment to produce risk for. So that's kind of the current direction of where we're taking this assessment platform that now allows us to do research in collaboration with schools all around the US and eventually all around the world. So I'll end there, and I think we have plenty of time for questions.